Okay, so this is chapter 24.3, amino acids and their polymers. We are on page 769. Key concepts. What is the general structure of an amino acid? Which functional groups are always involved in amide bonds between amino acids? What determines the differences in the chemical and physiological properties in peptides and proteins? How do enzymes affect the rates of reactions in living things? Vocabulary: amino acid, peptide, peptide bond, protein, enzymes, substrates, active site.
Want me to read connecting to your world? Yeah. Alright, connecting to your world. Many people are lactose intolerant, which means that they cannot digest milk or products containing milk. These people cannot digest milk products because their bodies do not produce enough of the enzyme lactase to digest lactose, the sugar found in milk. The undigested lactose causes bloating and stomach upset. Some people with lactose intolerance can enjoy milk products if they take a pill containing lactase before eating. In this section, you will learn what enzymes are and what function they serve in the body.
Amino acids. Amino acids have a skeleton that consists of a carboxyl group and amino group, both of which are covalently bonded to a central carbon atom. The remaining two groups on the central carbon atom are hydrogen and an R group that constitutes the amino acid side chain. Peptides. The amide bonds between amino acids always involve the central amino and central carboxyl groups. The side chains are not involved in the bonding. Proteins. Differences in the chemical and physiological properties of peptides and proteins result from differences in the amino acid sequence. Enzymes. Enzymes increase the rates of chemical reactions in living things.
How enzymes work. Coenzymes.
Let's look at pics, guys.
Okay.
Okay.
So demanding.
Where'd it go? What number? Where is this? Oh, figure 24.6. Peptides form three-dimensional shapes. A. This is a representation of amino acids in a peptide chain. B. The chain may coil into a helix. C. Two peptide chains may become arranged in a pleated sheet-like structure. Applying concepts. What types of bonds determine the three-dimensional shape of a protein?
Figure 24.7. The three-dimensional structure of monoglobin, the oxygen storage protein of this muscle tissue, is shown here. Most of the peptide chain of monoglobin is wood into helixes, right? Okay, myoglobin also contains a non-protein structure called heme. The heme groups is shown as a disc in the monoglobin structure. Heme contains four linked rings with an iron 2, iron Fe2+ at the center. Molecular oxygen binds to the heme iron.
Figure 24.8. A substance fits into a distinctively shaped active site on an enzyme. Bond breaking occurs at the active site to produce the products of the reaction. Predicting. What would happen if access to the active site were blocked by another molecule?
Figure 24.9. The color-enhanced scanning electron micrograph shows HIV red particle infecting a human white blood cell orange. The diagram models the enzyme HIV protease. The green and yellow ribbons trace the two peptide chains of the enzyme. A substrate molecule, purple, is embedded in the active site formed between the two peptide chains.
Okay, for notes, guys, you're going to draw that diagram on page 769. It's really short and sweet, so that's good.
That little tiny peewee thing.
Yeah.
Wait, what diagram?
That little diagram kind of towards the bottom of the page on 769.
Oh, okay.
Maybe you should listen next time.
He was just.
I know, and it's annoying.
That's very rude. Just saying.
Okay, let's not have a big debate over it. Let's just let it go.
You should
Slap each other and get it over with.
I wrote side chain, so side chain 4.
The kids come home crying. Just slap yourself and get over it.
The first one is the first big bold one.
Doubtful. I'm sorry. He was in charge of our cues.
So every time you see a COOH, you know it's a
Carboxyl.
carboxyl group. Correct?
Oh.
Okay, so finish. Tell them what we're doing tomorrow.
Okay, for notes, and underneath your notes, put notes section 24.3 RQ 1 and 2, page 773.
We'll do that.
And we'll do that tomorrow.
All right. Good deal. Get it there.
Where are you going? Just run away.
We left off on the RQs, so this is notes, section 24.3, RQ 1 plus 2, page
773.
Okay.
And number 1 is, what are the 4 groups that surround the central carbon atom in an amino acid?
Hey, what?
773.
And the final way to write down is on page 760.
And the one we write down is on page 765. It's the
Nine.
first
69.
69. It's the bolded one.
Oh my gosh. The whole thing?
Yeah.
Yeah.
You don't want to shorten that up a little bit?
We couldn't, we tried.
Same thing I said yesterday.
Anything else?
So capital A. Amino acids have a skeleton that consists of
Carboxyl.
a carboxyl group and an amino group, comma, both of which are covalently bonded to a central carbon atom, period. The remaining 2 groups on the central carbon atom are hydrogen, and R group constitutes the amino acid side chain, period.
So does anyone want to read back to us so the whole class knows?
Let them write first.
Yours is supposed to do that.
Oh, never
Not
mind.
us.
Let them write first. They're still working on it.
Oh, never mind. I lied.
Why does that sound?
It's because I'm writing really hard, I guess. So the whole table shaking.
Is that why that table's shaking?
Yes. Yes. I'm sorry.
Who's reading it?
Okay.
Dakota, read it back.
So mean.
Amino acids have a skeleton that consists of a carboxyl group and an amino group, both of which are covalently bonded to a central carbon atom, period. The remaining 2 groups of the central carbon atom are hydrogen and an R group that constitutes the amino acid side chain.
Okay. And number 2 is, which functional groups are always involved in an amide bond?
It's on page 760.
Amino bond. Amino bond.
And it's the first bolded on page 770.
I knew it.
Thank you.
So capital T, the amide bonds between amino acids always involve the central amino and the central carboxyl groups, period. The side chains are not involved in the bonding, period.
I'm still uninvolved. What is involved?
the back, Dakota.
You're so mean.
Do we write the side chains are not involved in the bond?
Yes.
Okay. Bring it back to you guys. Capital T, the amide bond between amino acids always involve the central amino and central carboxyl group. Period. Capital T, the side chains are not involved in the bonding. Period.
And we are done.
Be
These blocks make me